Good afternoon. Welcome back to Virology. Next two lectures, we're going to talk about preventing viral disease. Today, we'll talk about vaccines. Vaccines prevent viral disease. They don't prevent infection. I want you to remember that. True or false? Vaccines prevent infection. False. Everyone thinks they prevent infection. Now you know they don't. They prevent disease. Vaccines are the best proven defense against viruses. Not vitamin A, not cod liver oil, not a healthy lifestyle, blah, blah, blah. Vaccines are the best proven defense. They mobilize our immune system to prevent virus disease through making immune memory. So here's a lifespan or a life expectancy of humans, men and women, from 1900 to just past 2000. You see it steadily increased from about 50 years to close to 80. This is due to public health and vaccines and other medicines. That dip in 1918, do you know what that was? Spanish flu, yes, flu, the great influenza, let's call it. We won't blame it on a country. And if we had had vaccines, it would have made a difference. For example, here is a life expectancy, 1980 to 2020. Total population, Latino, white, and black. You can see steadily rising, and then suddenly 2020, boom, a crash. Remember, that's the first year of the pandemic where we don't have vaccines. So again, another pandemic impacts life expectancy. It dropped 1.5 years in 2020. Now, the way vaccines work is they stimulate a protective immune response. You know this already. This is just a diagram of the immune response with antibodies or T cells on the y-axis, because vaccines can do both, time in days. So we have this is actually for infection, but it could be for vaccination as well. You have your first infection, within 2 weeks, you make antibodies and T cells. Those then go down to a baseline level within time. And then if you are infected later on, you have a very rapid response in antibodies and T cells because you have memory cells and you have mild or inapparent infection. It really depends. But even memory does not prevent infection. It can't. Think about it. You get infected. Takes a few days for the adaptive response to kick in. During that time, the virus is replicating. Even under the best conditions, you're going to get infected. If you are young and healthy, you will have moderate disease. If you are older with other problems, you could have severe disease. You could go to the hospital still with a memory response because you have other issues. And that's why we have, still have up to 1,000 deaths a week from COVID. Most of those are in unvaccinated people, but most of them are elderly as well. So that's how vaccines work. So the first vaccine was a vaccine against smallpox. I think I've told you this story before. In the 11th century in China, they practiced variolation, which meant they knew that people who got smallpox never got it again. So they would take the pustules and scrape it into other people to give them some kind of protection. In 1796, Edward Jenner in the UK formalized this. He noticed that milkmaids, they milk the cows, they get cowpox from the cows. They get lesions on their hands. And they never got smallpox. Now remember, at this time, there's no knowledge of viruses. But he said there's some infection going on and cowpox can prevent it. So he did an experiment with one child. He immunized the child with cowpox, pustules from a milkmaid. He waited 2 weeks. He must have seen this graph, right? He waited 2 weeks. And then he challenged the boy with smallpox. I mean, braveness on both sides, right? Because I'm pretty sure that none of us would let anyone do that today. And you shouldn't. It's a challenging experiment that is not ethical. The boy was protected against smallpox, and then from that point on, vaccination spread throughout the world. In 1885, Louis Pasteur in France coined the phrase vaccine. He developed a rabies vaccine. He named it in Jenner's honor because vacca, as you may know, is Latin for cows, so it's a vaccine. And so that's where we get the name vaccines from. We're honoring Jenner. And the earliest after rabies vaccine, the next vaccines were in the 1930s, the yellow fever vaccine. The first influenza vaccine was in the 1930s. By the way, RFK Jr. says the 1918 pandemic was caused by the flu vaccine. Have you heard that? There weren't any flu vaccines until 1930. But nobody knows this. Now you do. All right? So you got to call him out. I'm equipping you to go forth into the world. Anyway, the smallpox vaccine is given on this bifurcated needle. And you put a drop of vaccine there. You scrape it into the skin. You actually make cuts. And the virus replicates. And the scraping causes inflammation, which helps get a good immune response. I still have my smallpox vaccination scar here because I was around before the virus was eradicated in 1979. However, if you're in the military, you will still be getting smallpox vaccine to this day. As soon as Jenner's vaccine arose, so did the anti-vaxxers, immediately. This is a woodcut from the era. It says here, the cowpox, or the wonderful effects of the new inoculation. The publications of the Anti-Vaccine Society. And these people are growing cows out of their arms where they got, here's Jenner inoculating people, and then they grow cows out of their, I don't know why this fellow has got it in his nose. But from the start, we have had anti-vaccine sentiments. Nevertheless, vaccines, especially large-scale vaccine campaigns, are very successful. We have two examples here, polio and measles. At the top, tens of thousands of cases of paralytic polio in the US. In the 30s and 40s, we introduced a vaccine in 1955 and a different one in 1962 and eliminated poliomyelitis from the US. At the bottom, measles. Hundreds of thousands of cases in the US before vaccine. Vaccine introduced in the 60s. Measles was eliminated in 2000. We are on the verge of losing our specification of having eliminated measles because it's coming back big time. And on the right is a graph that shows you lives saved by measles vaccination. So this would be, at the dark purple, the estimated measles deaths in the absence of vaccination. So this is in millions here, and here are the actual measles deaths with vaccination. So many, many millions of lives have been saved. It is not a trivial disease, as I've told you. Vaccines are part of our existence. We immunize almost everything. Children, adults of all ages, we immunize our pets. We even immunize wild animals. We immunize wild animals against rabies by dropping bait into the forest, which has in it a rabies vaccine. And because of immunization, childhood diseases are now rare, and that's part of the problem. Parents say, there's no polio, there's no measles, there's no any of this. Why do I have to vaccinate my kids? Unfortunately, vaccines are not yet part of low and middle income nations. As you saw, the measles, mumps, rubella vaccine isn't given sufficiently in those countries, and we have a lot of disease. We even immunize fish, yes. There's the obligate cute kitty, yes. But fish, because they can get viral diseases. Unfortunately, vaccine hesitancy has grown since the day of Jenner. It really grew big time in the 50s with the first polio vaccine. But All these things are what people say to explain why they're not getting vaccinated. Viral diseases are a thing of the past. Polio is long gone. Herd immunity has not been proven to work. I've met physicians who tell me that. Measles is a trivial kid disease. Kids should get infected naturally. Okay, 1 in 1,000 encephalitis. That makes a lot of sense. I'm not injecting anything into my body. Ha! Ha! Liars. Vaccines cause autism. Not true. They don't cause multiple sclerosis. We don't have time. When you have these kinds of attitudes, you have problems with large-scale vaccination programs. And, you know, the number of people who get vaccinated is a Venn diagram of confidence, complacency, and convenience. In some cases, medical exemptions to vaccination are indicated, but they should not exceed 1% of the US population. But they usually do. In some counties in Texas, they exceed 50%. Why are these physicians giving unnecessary medical exemptions? I do not know. You know, when you become a doc, you take an oath to do no harm. And so I don't get this. I don't get why people forget that. So lots of physicians give medical exemptions throughout the country. And there are also religious and personal exemptions. Many states have eliminated those. And of course, many states have school requirements for vaccination. All the states have school requirements for vaccinations of some kind. I did an interview a long time ago with 2 physicians who were involved in vaccination. Very, very good. Worth a listen. Vaccine programs will work only if people believe their value. And we have a number of cases in the US. This is a map of measles outbreaks tied to religious communities, unvaccinated members of religious communities, or not tied to religious communities. This is a bit old. It's not up to date. But you can see with the yellow, these are all the states with outbreaks of measles. In the red here, New York and North Carolina and Texas, these are communities where the religious leaders encouraged them not to get vaccinated. So big outbreak of measles in Brooklyn in an Orthodox Jewish community, an outbreak in a Hare Krishna community, and here in Texas, an outbreak where the senior pastor was critical of measles vaccination. Do you think the senior pastor has an MD or a PhD in public health or immunology or virology? What the hell does he know about vaccination? He knows nothing except what the anti-vaxxers pass on to him. But unfortunately, his congregation listens to him, and that's part of the problem. And now we're involved in a big outbreak in Texas. Here's the cases of measles in the US since 2023. You can see it's the biggest outbreak in years. It's spreading to other states as well, and there have been 3 deaths associated with it. And part of the problem is the misinformation business. Joe Mercola, he's a physician in Florida. He is a millionaire. He runs the most popular alternative health and dietary supplement website in the world. He has 1.7 million followers on Facebook, Twitter, YouTube, and he distributes misinformation. And he says, instead, buy my products and they will take care of you. Well, he's making money off of this. This is the basis for his misinformation. Early in COVID, he said, he promoted on his website, Stop COVID Cold. He offered hydrogen peroxide and quercetin as treatments for COVID. Unproven, and to this day, don't do anything. He said COVID vaccines were a medical fraud. They don't prevent infections. Do vaccines prevent infections ever? No, they do not. They don't provide immunity. Really? They don't stop the spread of disease. Really? All lies, by the way, all lies. He says the vaccine alters your genetic coding, turning you into a viral protein factory that has no off switch. Lie. It's a lie. It's not right. You're not a protein factory. The mRNA vaccines go in for a period of time and then they are gone. It's very clear, but people think he knows something because he's a physician. Then we have RFK Jr., who perhaps is even worse, and now he's head of Health and Human Services. What does that mean? CDC, FDA, NIH are all under his guidance. He believes in miasma. He thinks there's no such thing as infectious agents. Bad air causes disease. He says vaccines cause autism, despite over a dozen studies that show they don't. He claims no vaccine is safe and effective. He claims the measles vaccine kills people, causes deafness and blindness, and causes measles. None of that is true. It hasn't killed a single person. He claims that polio vaccine has killed more people than it saved. Lie. He claims the COVID vaccine was the deadliest ever made. Lie. You cannot believe a word he says. Yet, he was confirmed by the Senate. And he said all these things in the Senate. And now we have this Texas measles outbreak, which began last October, but has gotten worse under his guidance. It happened in a homeschooled Mennonite community in West Texas. So these individuals do not have to have school vaccines because they homeschool. First death of a child, February 26th, the first measles-related death in 20 years, completely preventable. RFK Jr. says it's not a big deal. There are always outbreaks. We have measles every year. He did not advocate vaccines. He said they're a personal decision. Instead, try vitamin A. It works miracles. It doesn't. It doesn't do anything in a community that's fully nourished. And so, but everyone decided to try vitamin A in Texas, and a bunch of kids went to the hospital with liver damage, which is one of the outcomes of overdosing. Vaccination, and this is the last slide on this topic. Vaccination is not a personal decision. This is a quote by Don McNeil in the Washington Post. RFK Jr. needs to stop saying vaccines are a personal choice. They're not. Speed limits are not a personal choice. Paying taxes isn't a personal choice. Sacrifices are what make us a nation instead of 340 million selfish hunter-gatherers. Each of us gives up some personal freedoms so we all stay alive. And that's the key. You give up some personal freedom, you get vaccinated, maybe you don't want to, but you do that for everyone. And there are a lot of people in this country who apparently don't feel that way. And I'm very sad about that, because in many other countries, people will do it. But this is the US, right? Everyone's a cowboy of some kind. Okay, there are two kinds of vaccines: active vaccines and passive vaccines. An active vaccine is mostly what we'll talk about today. We put some kind of the pathogen into you, you make an immune response, you get memory, you prevent disease, okay? Gives you long-term protection because of memory. Passive vaccines, we give you now antibodies typically, or you could give immune cells or even T cells, but right now it's mostly antibodies, and that will prevent disease, and it's short-term protection. A very well-known one is the rabies passive vaccine. This is it here, rabies immune globulin. These are antibodies from people who have been vaccinated with rabies vaccine. If you get bitten by a rabid animal, they will inject this at the bite site to help neutralize virus and prevent its spread into the CNS, but you also need to get a rabies vaccine. Short-term protection, antibodies don't last forever. Now, when you are incubating in your mother, you're getting a passive vaccine, a natural passive vaccine. Here's a graph showing the fraction of antibodies of adult value with time. So here we have conception, and then 3 and 6 and 9 months, and birth. As you can see, IgG is slowly being transferred from the mother to the baby. These are serum levels of antibodies in either the fetus or the baby. They peak at around 8 months, and then at birth, they start to drop because they have a half-life, and you don't make your own yet. It takes you a while to make your own, as you can see here. After about 6 months, they're gone, but then yours start to kick in IgM, IgG, and IgA. So you're getting a passive immunization from your mother, and you don't have any choice. You have to accept it. There's a great example of passive therapy with convalescent serum. Back in the 1960s, there was an outbreak of Lassa virus in Nigeria. And this nurse who was working there, American nurse, she got the disease, but she recovered. They actually brought her to Columbia Presbyterian Hospital and treated her there. And so while she was there, they took her blood and they made serum from it and kept it. Jordi Casals was a virologist at Yale working on the virus. He infected himself. They brought him to Columbia and gave him serum from Penny Pinio, and he survived. For a very good example of using convalescent serum. Now, COVID-19, we've used it. I've talked about this before, but remember, we take people who have recovered, take out blood from them. We purify plasma, which is the liquid that remains when we remove the cells, and we prevent clotting. And we can give these to people to either prophylactically to prevent disease or therapeutically after infection to prevent disease. And then many monoclonal antibodies were developed, which are also passive therapy. You maybe remember these interesting names, etsevimab, and bamlanivimab, or just Bam Bam, we used to call it, and others that target different parts of the spike protein and prevent attachment to ACE2. So these are passive antibody therapies. After a while, no one made these anymore because the virus kept getting resistant to them. Probably no one here knows what Bam Bam was, who Bam Bam was, right? You know? Yeah, very good. It was one of the two kids, right, in Flintstones, Bam Bam. So we used to call this monoclonal Bam Bam because we couldn't pronounce it. All right, first question is, passive vaccination, A, takes place between mother and developing fetus. B, involves giving antibodies or convalescent plasma to the patient. C, confers short-term protection against disease. D, although administered intravenously, may still protect against respiratory disease. E, all of the above. Everybody's done. Look at that, 13 out of 13. The answer is all of the above, which most of you got. So mother and fetus for sure. Antibodies or plasma, I gave you examples of that. Short-term protection, yep. And yes, we gave the COVID antibodies intravenously, but they protect against respiratory disease. So obviously the antibodies can go from the blood to the respiratory tract. Some of the requirements for an effective vaccine: you have to make the right immune response. You have to know what the right immune response is for a particular virus. Remember, Th1 versus Th2. Th1 being making cytotoxic T cells, Th2 making antibodies. We didn't actually know for COVID. We took a guess. Secondly, you have to be protected against disease caused by the pathogen. It's not enough to make a response. So you can immunize people with the vaccine and get antibodies or T cells, but you have to be protected against disease. And so you need to do a clinical trial to demonstrate that. In addition, the vaccine has to be safe. It cannot cause the disease you're trying to prevent, and it has to have minimal side effects. So they're extensively tested for that. It has to induce protective immunity in the population, protection against disease. Protection needs to be long-lasting, and it needs to be cheap. The WHO would like all vaccines to cost less than $1. It has to be genetically stable. It cannot change. If it's an infectious attenuated vaccine, it cannot change. Sometimes it happens, though. Storage conditions. Many vaccines require low temperatures. You may remember when the COVID mRNA vaccines first came out, they needed to be dry iced. And they were figuring out how to ship all these vaccines on dry ice to all different locations. But most of the time, you just need to keep them frozen. Ideally, it would be nice to keep them at 4 degrees or room temperature. And finally, delivery. Most vaccines are delivered by needle, and people don't like needles. They won't tell you that, but they don't like them, and they don't like seeing their kids get needles. So it would be nice if we could have more oral vaccines or some other delivery methods. This is an interesting invention that was made under WHO supervision. This is a kerosene-fired refrigerator, basically, that will keep vaccines frozen, and it can be carried into the field, as you can see there. Okay, let's talk about the different kinds of vaccines, and I'll give you an example of some of them. So in all cases, we start with the virus that causes some disease that we want to prevent. And then we can do all the things shown below to make vaccines. We can make infectious vaccines. These are called attenuated or replication competent vaccines. We can inactivate the virus particles and give them to you inactivated. We can fractionate the virus and give you purified protein. Or in the modern era, we can clone the genes that are protective, and we can produce the protein and either make a subunit vaccine. Now it's a recombinant subunit vaccine, or sometimes the protein will assemble into virus-like particles, empty capsids. We can make DNA or mRNA vaccines with the right gene, or we can put the gene into vectors. You saw examples of these for COVID, multiple vectors being used to deliver the spike protein gene, and of course, mRNA vaccines encoding the spike protein. These are the viral vaccines licensed in the US. Here's the virus kind of vaccine here, and this would be what it's used for in the schedule. And you can see not all of these vaccines are for everyone. Here, for example, Adeno is for military recruits. Hep A is just for travelers. Hep B, of course, universal in children. Influenza should be for everyone. Japanese encephalitis, just for travelers to places where the virus is endemic. And then we have measles, mumps, rubella, universal vaccination for children, polio, and so forth. Rabies, only if you get exposed, or if you're a veterinarian or you work with animals, you should get it ahead of time. And also yellow fever, you don't need to get it here unless you're traveling somewhere where the disease is present. Then, of course, we have an Ebola virus vaccine, which is licensed. And we have 2 mRNA vaccines for COVID and a protein-based vaccine for COVID, Novavax. Now, this one has never been licensed. It's still being used under emergency use authorization. If you have an emergency situation, the FDA can give approval if you give them the right data, because licensure takes a long time. And so since we were in an emergency situation, they gave EU approval to multiple vaccines, and that Novavax still hasn't been approved for various reasons. So let's talk about inactivated vaccines. You take the virus, you treat it in a way to destroy infectivity. And there's chemicals you use like formalin, propiolactone, or detergents. You eliminate infectivity, but the antigenicity is not compromised. If you inject this into someone, they will still make antibodies and T cells to the right epitopes. So an example is the polio vaccine, inactivated polio vaccine. Now, poliomyelitis is a disease that was present for many years, still is. Here's where the name comes from, polio and itis. And from a 1959 textbook of medicine, the definition of poliomyelitis, a common acute viral disease characterized by a brief febrile illness with sore throat, headache, and vomiting, and often with stiffness of the neck and back, and in many cases, a lower neuron paralysis develops in the early days of illness. And so poliomyelitis became epidemic around 1900, as we've discussed before, and many outbreaks over the years. The post-World War II babies, the baby boomers, contributed to huge numbers of polio cases. Here we have 50,000 here at that peak. And then 2 polio vaccines were developed, and they led to the elimination of poliomyelitis in the US. In many places, when you got polio, you were put in an iron lung so you could breathe. So sometimes the chest muscles are affected or the nerve centers. And so we didn't have intubation back then. So we put you in an iron lung, which would fill with air, compressing your chest, and then the air would go out and your chest would expand, so you would breathe in. Very gentle way of giving air In fact, many physicians said, I wish I had iron lungs during COVID because intubation is very rough, putting a tube down into your lungs. The virus, of course, is poliovirus, an RNA virus, icosahedral capsid with a plus-strand genome. Now, FDR had polio in his 20s. He couldn't walk. He has leg braces here. He used canes. He used a wheelchair. This is one of the few pictures of him, though, that anyone saw. He raised the money to fund the development of both polio vaccines. It was not funded by the US, NIH. There was no NIH at the time. And the first vaccine that was developed was the inactivated vaccine, poliovirus treated with formalin to destroy infectivity. It was given a clinical trial in 1954, the biggest ever done, 1.8 million children. It had about 50% protection, and it was licensed the same day. 50% protection. I remember at the beginning of COVID, the FDA had said, if we get 50% protection, we will license COVID vaccines. And this is what that was based on. Big headlines throughout the world, as you can see. It was a big deal. Now, unfortunately, a few weeks after the vaccine was released, some kids developed polio because they got batches of vaccine that were not properly inactivated. And they were made by a company called Cutter Labs. There were many lawsuits about this, and this really amplified anti-vaccine sentiment because you have to think parents were bringing their kids to get vaccinated. They were so scared of polio. They put their trust in it, and then a few hundred kids get polio from the vaccine. It was really unfortunate. Well, they did fix it, but the damage was done, and anti-vaccine sentiment persists to this day. So that vaccine was introduced in 1955, and it cut the cases to a few thousand a year. The way it works is you get injected intramuscularly, you make antibodies and T cells in the blood, and when you get the virus, it's brought in by fecal-oral contamination, reproduces in your intestine, and gets into the blood via the lymph. And there the antibodies will neutralize virus and prevent disease. You don't prevent infection because you're still shedding lots of virus from the gastrointestinal tract. Now, about 1% of infections in the absence of vaccine are paralytic. So this prevents that because it prevents the virus from getting into the central nervous system from the blood. So this is an inactivated vaccine that we use to this day in many countries throughout the world. Another inactivated vaccine is against influenza virus. Remember, an RNA virus enveloped with a negative stranded segmented genome. And the key proteins are hemagglutinin and neuraminidase, which are membrane proteins in the virus. And the protection against disease correlates with antibodies against those proteins. We have many deaths each year, as I showed you before, due to flu, and that's why we make a vaccine. It's grown in eggs. It's then formalin inactivated and then injected. We make about 100 million doses a year in the US. It's about 60% effective in healthy children and adults less than 65 years of age. As I said, protection correlates with antibodies against HA and NA. There have also in recent years been developed vaccine grown in cell culture. One of them is called flu cell vax, if you don't like the idea of eggs. So the problem with the flu vaccine is that the envelope proteins, especially the HA, changes every year, and we've talked about this. And so some years we have to change the vaccine every year to match that. And so what is done is they have a vaccine strain that grows very well in eggs, and they determine which hemagglutinin and neuraminidase you need in the vaccine, and they make reassortants where they swap out the HA and NA from the high-yielding viruses and use those to grow them in eggs. And every year, so this year, for example, the flu vaccine contains an H1N1, an H3N2, and a B Victoria lineage viruses. So it's a trivalent formulation. But here's how you have to deal with influenza vaccine selection. There's a global network of laboratories that collect data on what is circulating, what kinds of flu viruses and their antigenic makeup and so forth. And they do surveillance all year round. So that's the Global Influenza Surveillance Network, run by WHO. And then they make a recommendation early in the year. They just made one a couple of weeks ago. And fortunately, the US CDC accepted it, and they're going to do it as well. So in the early part of the year, you select the strain, you make your reassortants, standardize the antigen, look at the potency, formulate, test, and package. And then starting in September or late August, you can vaccinate for the flu season, which begins shortly afterwards. And so this is a year-round process. Some years you have to change it, and some years you don't, depending on what's circulating. And some years you get the right combination, and some years you don't. And it can all be because of one amino acid change in the hemagglutinin. These colored areas are the antigenic sites, and the ones at the tip, one amino acid change can impact antibody reactivity. So you have to look at that and make a decision. And as I said, some years it matches and some years it doesn't, and we have a lot of flu. Next question. Which statement about inactivated vaccines is wrong? Incorrect. Chemicals can be used to inactivate infectivity. They do not replicate. They can be dangerous if inactivation is not complete. Antigenic variation can make them ineffective. None of the above are incorrect. All right, how did we do? None of them are incorrect, but not all of you got that. So chemicals can be used to inactivate, that's correct. They do not replicate, that's correct, they're inactivated. They can be dangerous if you don't inactivate them, and antigenic variation can make them ineffective. Yeah, flu vaccine, inactivated flu vaccine, we just talked about, so none of them are wrong. Okay, next, subunit vaccines. Here, we can do it a couple of ways. We can take the virus and break it up and purify components and immunize with them, or we can say, the spike gene is what we need to immunize with. We get the gene for the spike protein. We can produce the protein or make a virus-like particle. So in the case of some viruses, they will assemble into virus-like particles. In other cases, they would just be single proteins. So let's have a look at how this works. There's a wonderful vaccine called Shingrix against varicella-zoster virus. This is the virus that causes chickenpox in kids. And then when you're 50 or older, remember the virus goes latent in your ganglia. When you're 50 or older, it reactivates and gives you shingles, a rash. And so this vaccine is for people who had chickenpox as children. And so you're vaccinating to prevent shingles. And what it is, this is varicella-zoster virus. It's a herpes virus with many glycoproteins. They took one of the glycoproteins, recombinant GE, and made recombinant protein in cells in culture. It's mixed with an adjuvant, which we'll talk about in a moment, and then injected. And this is nearly 100, in the clinical trial, this was nearly 100% effective at preventing shingles. It's now being evaluated to replace the childhood chickenpox vaccine because it's so good. And we have an HBV vaccine, which prevents cancer. It's the hepatitis B virus surface antigen produced in yeast. And the surface antigen is present here in the particle. Remember, it's got an icosahedral capsid with an incomplete double-stranded DNA genome. It's got a membrane around it, and in it is the surface antigen, which, when expressed in yeast, assembles into empty particles. And these, when injected, are very effective at preventing cancer. We have human papillomavirus vaccines that are also subunit vaccines. Now, papillomaviruses cause warts. There are over 170 different kinds of papillomaviruses, many different kinds of warts as well. Some of them are transmitted sexually, and they are the most common sexually transmitted disease in the US. You can see a breakdown here of men and women, the percent who are infected with these viruses. Some of these viruses just cause low-risk genital warts, the sexually transmitted ones, which are not a concern. Other types are at high risk for cancers. Cervical, vaginal, penile, anal, and oropharyngeal cancers in particular. We see about 30,000 of those a year pre-vaccine in the US, mostly types 16 and 18. Nearly half of Americans are infected with these genital HPVs, which are types 18 to 59, and a subset of those can cause cancers. And so a vaccine was developed to prevent cancer, and what was done was to take the capsid protein, express it either in yeast or in insect cells. You get virus-like particles made. These are used to immunize you, and they make antibodies that protect this cervix from disease. And these are the different kinds of vaccines that are made by different companies. Gardasil is produced in yeast, type 6, 11, 16, and 18. And Gardasil 9 has 9 serotypes. Cervarix is made by GSK, 2 types produced in insect cells. And these have to be given before you become sexually active. And if you do so, you can get 100% prevention of cancer. What age is that? I would say 12 years old is the latest that you should give it to your kids. And many parents don't want to give their kids this vaccine because they claim it will make them sexually active. I used to talk at high schools about this. But when I said that, everybody laughed because nothing will make you sexually active except hormones, not a vaccine. So, and actually studies have been done which show that kids don't become more sexually active after getting vaccinated. This can save their life, so they need to get it. And of course, RFK Jr. is suing companies, Merck in particular, over this vaccine. There's nothing bad that this vaccine does to you. It prevents cancer. So a subunit vaccine has pros and cons. So recombinant technology is fast. It's really, really fast to make the vaccine. There's no viral genomes or infectious virus, so no potential infections. But they're expensive, they have to be injected, and they're poorly antigenic. And that's because they are not viruses that replicate. They don't cause inflammation. Remember, when a virus kills cells and causes inflammation, you get really good adaptive responses. If it doesn't kill cells, you get poor adaptive responses. So what do we do to make up for that? We add adjuvants to cause inflammation. So subunit vaccines that don't cause inflammation, we add adjuvants. And we have a number of adjuvants that we use. You can see that these are the ones licensed in the US and Europe. Alum, AS1, AS4. This is the one in Cervarix. This is the one in Shingrix, AS1. MF59. And this one, for example, AS1 is a toll-like receptor 4 ligand. It stimulates inflammation. So the main way that these work is by stimulating inflammation, but they also keep the antigen at the injection site longer, slow release from the injection site. So they're very useful. But of course, the anti-vaxxers say that they cause all kinds of problems, which is not true. They don't. So what kinds of new vaccine technologies might we hope for? One of them that's really cool is this microneedle patch, which is that's someone's finger there. And it's a tiny square with little needles in it made of plastic. And you saturate it with vaccine and you put it on the skin with a Band-Aid. You don't need a needle and it doesn't hurt. And so these are being tested for various vaccines because I think that would really help in many situations if you're afraid of needles or in places where there's no one who can use the needle. And then making vaccines thermostable. You know, as I said, the problem is you have to keep vaccines frozen. And it turns out if you freeze dry them in sugars, you can increase their stability. So here is an experiment. We're just measuring influenza virus infectivity with time at 40 degrees Celsius, which is very hot, and 40 degrees with sugar. And you can see in 2 days, the infectivity is all gone without sugar, but if you add sugar, you can maintain some infectivity. So this requires some more research, but that's promising as a way to eliminate having to keep vaccines frozen. Another thing that's being worked on very furiously for influenza is to try and make a universal flu vaccine. You get once every 10 years maybe, and it protects you against everything, instead of having to get one every year. And the problem is that the epitopes that change in the HA are at the top, but the stem region has conserved epitopes that don't change, if at all. And so one strategy is to make recombinant hemagglutinins where the head is something irrelevant to humans, like an H9, which doesn't infect people. Well, actually, it does rarely, but not on a big scale. And then the stem would be from a human relevant virus like H1N1. So you immunize with this and you get antibodies to the head and some to the stem. And then you give a boost with a different protein where now you've replaced the head with another irrelevant head, HA, and then you keep the stem the same. And the idea would be that you get a memory response to the stem and it's going to be more robust. And you do that again with the third chimeric antigen. So you exchange the heads, retain the stem HA, and the antibody is redirected against what is otherwise an immunosubdominant stalk region. So we don't make a lot of antibodies against that. So these are all things that are being tested and may emerge in the coming years. Or maybe they won't. Maybe they'll fail and we'll have something else. Okay, our next question is, what are some requirements for an effective vaccine? Low cost, ease of administration, long-lasting immunity, minimal side effects, all of the above. All right, let's see how we did. Hey, 100% got it right. All of the above, that's right. Now we'll talk about replication-competent attenuated vaccines. These are virus vaccines where the virus infects you and stimulates immune response, but it doesn't cause disease. So that's what the attenuation means. It doesn't cause disease. And the idea is, if you have an inactivated vaccine, you have to give multiple doses to get a good immune response, right? First, second, third dose. But a replication-competent vaccine replicates in you and amplifies itself, so it's basically boosting you at the same time. So that's the theory. And one of the ways these vaccines have been made is by passing you take the parent virus, which causes disease, and then you pass it in different kinds of cells. And sometimes it takes hundreds of passages. So you take the virus, which grows well in human cells, then you can pass it here. It's in a non-human primate cell, and you do that over and over. And maybe with time, you fix mutations in the genome that allow the virus to preferentially grow in monkey cells, but it loses pathogenicity for humans. And so this approach has been used for yellow fever vaccine, poliovirus vaccine, and measles, mumps, rubella vaccine, among others. An example is FluMist. This is an influenza vaccine which is sprayed in your nose with a syringe. And the idea is that it reproduces in your upper tract and gives you immunity. This virus was developed to be cold adapted, which means it can't reproduce in your lung where the temperature is higher. So it preferentially stays in the upper tract. Temperature sensitive, which again prevents it from going down into the tract, and attenuated. And so when you put this in people's noses, the replication is confined to the upper tract, and you get protective immunity. And the way this works is there's a master strain that has all these properties, And then depending on which HA and NA you need each season, you swap it out by reassortment. So unfortunately, this is only made by one company and there's limited supply, and it doesn't actually work much better than the injected inactivated viruses. But that's the concept. The other one I want to spend more time on is the oral poliovirus vaccine, which developed by Albert Sabin. And this is a vaccine that you ingest. It reproduces in your intestine. It gets into the blood, and that stops there. It doesn't get into your CNS, and it gives you immunity. So this was introduced in the US in 1962. IPV had been used since 1955, but there were still a few thousand cases a year, although they probably would have gone away if they kept using IPV. But people said we should use a vaccine that more closely mimics natural infection. So we switched to OPV, and that got rid of poliomyelitis in the US after 1979, although just 2 summers ago, there was a case in upstate New York, again, in an unvaccinated person, which shows that unless you keep vaccinating, you will have these diseases. Now these viruses, there are 3 serotypes of polio. And Albert Sabin passaged each serotype in different ways, cell cultures, different kinds of animals, right? Many, many passages. And he would test them at each passage. He would inject them right into the spinal cord of a non-human primate and see if they caused paralysis. And eventually he got 3 strains, 1, 2, 3, which didn't cause any disease. They were subjected to clinical trial and licensed in the US in 1961. Sabin showed that the use of these vaccines in mass immunization campaigns could eliminate poliomyelitis or severely reduce it in a country. And so in 1988, the WHO said, we're going to eradicate polio by 2000 and stop immunizing. So is it 2000? It is 2024, and we have not eradicated poliomyelitis. And so let me tell you what's happened. First of all, can viral diseases be eradicated? Well, smallpox eradication was launched in 1967, and the disease was declared eradicated in 1978. That's the only major human disease to be eradicated. To be eradicated, you need 3 features. The replication must only occur in one host, people, not something else, like SARS-CoV-2 replication. So many animals, you can forget about eradicating SARS-CoV-2. Never going to happen. It's here, it's with us forever. Second, vaccination must induce lifelong immunity. And third, asymptomatic infections do not contribute to viral spread. So for smallpox, all 3 of these fit the bill, and so it was possible to eradicate. In other words, you saw a kid like this with smallpox, you went in and immunized everybody around her, and that stopped the spread because there were no kids asymptomatically infected. It's not the case with polio, as you will see. So where are we with the polio effort? 1988, 125 endemic countries. 10 years later, 40 endemic countries, which means the virus is still circulating there. 2008, 5 polio endemic countries, 191 polio-free. What about 2025? Well, before I get to that, let me tell you what is foiling polio eradication. When OPV was introduced in 1961, so here's a graph showing you in a line the total cases of paralytic polio from 1961 to 2003. And so this is the effect of IPV. But in 1961, we switched to OPV and eventually eliminated paralytic polio in the US. But what happened was, as we introduced OPV, we got cases of polio caused by the vaccine. Those are the bars. 1 paralytic case per 1.4 million doses, so that would be like 8 to 12 cases a year. And eventually, the last wild polio case was 79. Eventually, all the cases of polio in the US were caused by the vaccine. If the anti-vaxxers ever did their homework, this would be the one vaccine they should protest against. But they never did because they don't read anything. IPV doesn't do this. We should never have switched. How can it be good to paralyze kids at all? No, it's not. So in 2000, we switched back to IPV. No more vaccine-associated paralytic polio in the US. But guess what vaccine WHO is using for the eradication effort? OPV. OPV, you may remember, has mutations in the 5' non-coding region in this particular stem loop. Each serotype, 1, 2, and 3, has a single base change in this stem loop. When you feed this virus to kids, it reverts within a day. This is one child, DM, who got the Sabin vaccine. It has a U at 472. Within 47 hours, it's a C, which is from the virulent parent. And this child is shedding that virus for weeks. Every child that gets the vaccine sheds revertants. And 1 in 1.4 million develop polio. Probably they have some SNP affecting their immune response. But these viruses are virulent. If you put them in monkeys, non-human primates, they cause poliomyelitis. So imagine this scenario. We're immunizing millions of kids with OPV, and they're shedding OPV, which can cause polio. The worst culprit was the type 2 vaccine. This is a map of cases in October 23. These maps aren't made anymore because they want to hide it from us. WHO doesn't release these maps because it really looks bad, as you're going to see in a moment. So all the green dots are cases of vaccine-associated type 2 polio caused by the vaccine in areas where immunization dropped. Those countries all let immunization drop, and they got outbreaks of type 2. So WHO and CDC said in 2016, we're going to remove the type 2 OPV from the vaccine. That way we won't get any more type 2 vaccine-associated paralysis. So in 2016, they removed it. Big error. Disaster. Because there were still type 2 viruses circulating. We didn't know it. And here's what happened. We made human-made cases of polio. So here's before OPV2 withdrawal. In 2016, they take OPV2 out of the vaccine. These are the number of cases from 2010 to 2016. These are circulating vaccine-derived cases caused by the vaccine, 318 cases in 15 countries. We take the type 2 out of the vaccine, and look what we get on the right here. This is after OPV2 withdrawal from 2016 to 2023. 3,363 cases in 43 countries, including places where they hadn't seen it in years. These are all preventable. These are human-made cases of polio caused by the vaccine. How are they preventable? We could have been using IPV. But WHO will not stray from its goal of eradicating with OPV. You're never going to eradicate polio because you're using a virus that's infectious and shed into the environment. And that's why many countries use IPV, like US and much of Europe, Scandinavia, and so forth. So can we eradicate polio? No, we can't eradicate a virus for which 99% of infections are asymptomatic, but we can eliminate poliomyelitis, the disease, if we immunize globally with IPV. That's the solution. WHO is still using OPV in many countries, which I think is unethical. OPV has been delicensed in the US. You're not allowed to use it because it causes paralysis. So why is it okay to use in kids in other countries? It's not. It's unethical. And this is being perpetrated by WHO, which doesn't want to switch to IPV. Now, it could be that IPV is not the best vaccine. It's the only one we have. Maybe we should do some research. Well, WHO has declared that in June of this year, you can't do any more polio research, so you can't make any new polio vaccines. And we could use some new ones. They don't want you working on polio in the lab because they think it will get back into the environment from your lab. They think your lab is a threat. But guess what? Wherever you look in wastewater, you find poliovirus. It's throughout the New York metropolitan area. The CDC doesn't look in other states because they don't want us to know. It's in other countries. All these countries that had cases of polio, the virus is in wastewater. It's everywhere. So labs are not a threat. It's another stupidity by WHO to say labs are a threat. They say labs are a threat to eradication. They're not, because there's a lot of virus in the environment. And that's why the fellow in upstate New York 2 years ago, he wasn't immunized. He got infected by these type 2 strains that are circulating all over the place. So this is an interesting story where the vaccine is now causing the problem. Types 2 and 3 wild types have been eradicated. There's a little bit of type 1 circulating. But most of the cases of polio, as you saw, are caused by type 2 vaccine. Interesting situation, right? All right, last comments. We have a new class of vaccines now, right, developed during COVID, mRNA vaccines. 2 companies, Moderna and Pfizer, developed these vaccines. What they did is they took the spike protein gene, the mRNA coding region from the spike protein. They made a DNA copy. And then they did in vitro transcription with a phage RNA polymerase to make full-length mRNAs. Milligrams of mRNA. And these were made using modified bases that would not cause the innate immune response to be activated, which would cause clearance of the mRNA. They were then, those RNAs were incorporated into lipid nanoparticles, and then they're injected into your arm. So the target is the spike protein, right? The surface protein of the virus there, which we've talked about. And 2 prolines have been added to the C-terminus right here of S2. Sorry, this S2 right here. And that stabilizes the protein so it doesn't go into the fusion conformation, and it also makes it more antigenic. So that was the key to making a really good vaccine. So it's injected into your arm muscle. Your antigen-presenting cells, like dendritic cells, pick it up. They bring it to the lymph node. The APCs are making the protein. It's displayed on the surface. And you know the rest that happens. It's presented to T cells, and that initiates an adaptive response. These were produced in a year, really remarkable feat, and have prevented many, many COVID deaths. There are some side effects, besides the usual ones: fever, pain at the injection site. A very few number of people get myocarditis, which is not fatal and is treatable. The rate of myocarditis with infection with SARS-CoV-2 is much higher. So there's no reason not to get this vaccine, yet they're being vilified by the anti-vaxxers for reasons that are just not correct. Okay, that's a brief overview of vaccines which prevent disease. Next time we're going to talk about antivirals, which you can give after you're infected, right? So for vaccines, typically we give them before you're infected. What's the one exception? What vaccine do we give after you're infected? Rabies. It's the only one. The others we give before you're infected. But antivirals you can give when you're already infected.